So, we will continue with this facts analysis. So, here it is another set of figure which is little bit more details, as you can see. So, you have a flow chamber here. So, from flow chamber individual cells are being charged particles here, and in these charged particles they will go in the positive side and negative side. So, negatively charged particles will be attracted towards the positive deflecting electrode, and negatively and positively charged cells will be deflected towards the negative electrode, right. So, this is based on a very simple technique, and then this uncharged particles they will be going to this straight. Okay. So, these charged cells they will be collected at two different test tubes, and now let us see that how these results they look like. Okay. Okay, this is that hydroxyapatite 20% mullite particles. The eluate preparation and the particle preparation are the same as I have discussed earlier in case of the cytotoxicity experiments and single cell gel electrophoresis experiment. So, control means essentially this is like untreated cells. Okay. So, that HFOB cells, HFOB stands for human fetal osteoblast cells. HFOB cells when they are treated with H20M eluate, then after 12 hours what are the fraction of the live cells, fraction of the necrotic cells, fraction of the late apoptosis cells, fraction of the early apoptosis cells, how you can distinguish using that fax machine, and this is the results. Here you see that Pi stands for propidium iodide, it interacts with the DNA causing the red nucleus in case of the necrotic cells. Annexin, you know that annexin 5 is another dye, and then FITC is that fluorophore. So, annexin FITC they in combination they will stain differently if it is apoptotic cells. So, what are the different coordinates here? Q3, that means this one is for the live cells. Okay, Q2, that means this one diagonally up, that is for necrotic cells. Okay. Q4, that is for the early apoptosis cells, EA. Q1, that is for the late apoptosis cells. So, what is the pattern, evolution pattern you see? It is more like a sigmoidal curve type of patterns. You see that after this treatment cells are following this kind of pattern. Okay. Now, let me first tell you each data point here, you see that each data point corresponds to the information of one single cell. Now, after 10% eluate treatment, that means after 10% of the hydroxyapatite 20% mullite eluate, okay, what you see your live cell coordinate, this is your live cell coordinate, here you have most number of the cells, that means your most of the cells are live. However, you do see noticeable cells in the necrotic phase, also noticeable number of cells are in the early apoptosis, with a few cells they are entering in the late apoptosis stage. When you put this treatment as 50%, what you notice here that more number of cells are now going to the early apoptosis phase. Okay. And also similarly higher number of cells compared to 10%, if you see that concentration of the dots, right, concentration of the dots in the necrotic stage is much lower after treatment with 10% compared to the concentration of the necrotic phase here after 50% treatment. If you more see when they are treated with 100% eluate, you see more number of dots are here, and more number of dots means more number of cells are in the necrotic stage. Another important things if you notice the concentration of the dots in the early apoptosis coordinates, look at the slide here, and you yourself will be able to realize that if you increase the concentration of the eluate or particles from 10% to 50% to 100%, if you see the number of dots in the early apoptosis coordinates increases progressively from 10% to 50%, 50% to 100%. Clear? So, therefore, that more cell fractions of the total cell populations are increasingly entering into the early apoptosis stage when you are treating them with the progressively higher and higher concentration of the hydroxyapatite 20% eluate concentration. Is it clear? So, now you know how to read the FACS data, right. So, you have 4 different coordinates, and in these 4 different coordinates you have different dots. Each dot corresponds to one single cell information. Okay. So, you can quantify using the standard software that what is the total fraction of the cells that are now in the apoptosis stage, early apoptosis, late apoptosis, necrosis and all this stage. Now, what happens when you treat them for 24 hours? This is control, means again is the untreated cells. There is no treatment of the hydroxyapatite 20% moolite particles here. In case of the untreated cells, what you notice here that in the 10% concentration, your some cells fractions are going to the early apoptosis. 50% more number of dots means more number of cells are in the early apoptosis. 100% clearly more number of dots here, that means more number of cells in early apoptosis. So, there are two things. One is that your concentration of eluate, and one is your treatment time. So, with 24 hours you see that with the higher and higher concentration, more number of cells are going to the early apoptosis stage and necrotic stage. What happens in the 48 hours treatment? 48 hours you notice that really large number of cells are going to the necrosis stage, you see, and these numbers if you qualitatively compare, these numbers are more than what you have seen at the corresponding concentration after 12-hour treatment. Do you understand what I am saying? I am saying that let us now compare what happens after 10% eluate treatment, after 12 hour and after 48 hour. So, after 48 hour here you see that 10% 100% concentration more number of cells are going to the necrotic stage. That means, the severity of the cell death increases with increasing concentration or with increasing treatment time. Now, in the next few slides I will be showing you that how different molecular markers means different genes which are involved in the bone cell differentiation process. Bone cell differentiation means when it is going to live cells to apoptotic cells, right. Bone cells, live cells, your DNA will have certain arrangement, your plasma membrane will have certain function. In the apoptotic cells, your plasma membrane also will have different functions, and DNA will not be able to replicate and make the protein or synthesize the protein like in the live cells. Agree on this point? So, therefore, the ability to DNA to transcript and translate to make the proteins, or the ability of to DNA to go through the replication process, that will be different depending on the whether the cells are live or the cells are apoptotic or necrotic. Now, this is known as that QRT-PCR technique, which is known as Q stands for quantitative, RT stands for reverse transcription, PCR stands for polymerase chain reaction. Now, you can now follow, because you know that how from DNA how this RNA is produced and then these proteins are produced. Now, total RNA is initially extracted from the control. What is control here? Control means untreated cells. Okay? That means cells which has not received any treatment of the particles like 20% moolite particles. Okay. So, total RNA extraction, if you total RNA is extracted from the control untreated cell as well as H20M nanoeluates. H20M stands for hydroxyapatite 20% moolite nanoparticle eluates. Now, this RNA which is extracted plus reverse transcriptase enzyme. So, reverse transcriptase enzyme means if you want this reverse transcription process, reverse transcription means what? DNA to RNA is transcription, reverse transcription means RNA to goes back to DNA. So, that is why I often say that all these biological terms are very easy if you follow it in a logical manner. Okay. So, you require certain enzyme to be added to RNA in order for the RNA to go back to the DNA double helix pattern, and random hexamer primers for the RT-PCR. So, there are primers, I will come back to that. Now, you have to amplify the fragment of the BCL, BAX, CBFA, osteocalcin, collagen, beta actin 18S by qRT-PCR. Now, what is the meaning of this statement? So, up to the step 1 it is clear that you can extract RNA from the untreated cells as well as the treated cells. Step 2 is also clear. Now, reverse transcriptase means mRNA goes back to DNA, but you require certain enzyme to be added and some primer to be added. Now, these primers are essentially combination of the nucleotide. What is the combination of the nucleotide? You know, for DNA, what are the nucleotides that you require? A, T, G, C. So, certain sequence of the nucleotide you have to add to the RNA to make RNA go back to the DNA. Why? Because RNA has a different nucleotides, because RNA has AUGC, adenine, uracil, guanine and cytosine, whereas DNA has adenine, thymine, guanine and cytosine. Now, what is stage 3? Now, RT-PCR, what RT-PCR does, after you do this treatment, this RNA plus reverse transcriptase enzyme, then what will happen, that it will produce certain fragment of BCL, BAX, these are genes, okay. CBFA, they are genes, osteocalcin, this is another genes, collagen, that is another genes. These last two, beta actin and 18S, this beta actin and 18S, they are known as the housekeeping gene. What is known as the housekeeping gene? What is the name suggests? Name suggests is that housekeeping means if you want total nucleus to be in the order, okay. In the order means this nucleus should be able to replicate during the cell division process, and nucleus you want that all this chromosome and DNA to carry exactly the genetic information, then you require certain genes to be very active in the cells. So, beta actin and 18S are the two genes independent of the cell type, they are always active in case of the live cells. You understand what I mean? I repeat, independent of the cell type means it does not matter whether it is a fibroblast cells, osteoblast cells, endothelial cells. You will always find beta actin and 18S, these are two different types of genes which are present in the nucleus, so that the normal functionality of the nucleus can be maintained. So, in other words, it is typically known as that ubiquitous gene, that means it is present in all cell type. And now, why do we require to amplify the fragment? What happens after this RNA transcription, RNA reverse transcription process, the DNA that will be produced and this BCL, CBFA, osteocalcin and collagen, these genes that are expressed means the amount that will be formed of all these genes, that will be very smaller in number or smaller in quantity. You have to amplify it, otherwise you cannot distinguish between the amount of BCL gene, amount of CBFA gene, amount of osteocalcin gene. Are you getting my point? You have to amplify their expression, you have to amplify. So, what the way they are expressed, you have to amplify it to 10 times, so that you can distinguish the difference between these two. Okay. So, for example, suppose if I say that the strength of certain two materials, they are like 1.2 gigapascal and 1.1 gigapascal. Okay. So, you may see that 1.2 gigapascal, 1.1 gigapascal, it is 0.1 gigapascal difference. The moment I say the strength of two materials is 1200 MPa and 1100 MPa, then you can immediately say that there is a 100 megapascal difference. That means 100 megapascal is large. So, there you are going down in the unit. So, to amplify the difference. Okay. So, in here you have to expression that BCL BAX, these are like different genes, and these genes BCL, that is a class of genes, this is the BCL2 is a class of genes which are activated in osteoblast cells. Now, in BCL family you have the different genes, that is BCL-XL genes, they you have called BCL-XS gene. Now, BCL-XS is the genes which are called pro-apoptotic genes. Pro means what? It will help in the apoptosis process. BCL-XL is called anti-apoptotic genes. Anti-apoptotic means these particular genes, if it is expressed more, that means cells are more live or more vital. That means cells are not going to the apoptosis process. Is it clear? So, apoptotic, okay, apoptotic process. So, I repeat, so BCL-XL, BCL-XS and BAX. So, these are three genes BCL-XL, BCL-XS and BAX. These are three genes, they all belong to the family of the BCL2 family of the genes. Okay. Now, this BCL2 family of the genes, they are important for this toxicity process, apoptotic process. However, in this family of gene, the first gene, this is anti-apoptotic. The two other genes, these are like pro-apoptotic. Pro-apoptotic means if these genes are expressed more, that means cells will undergo definitely to the apoptosis process. If the first gene is expressed more, then cells will be more live cells. Okay. Okay. Now, so therefore, in this first slide, what should be clear to you, that QRT-PCR process is helpful to identify the molecular markers. Molecular markers means the genes here, genes of interest, which are helpful or which are responsible for the apoptosis process. FACS machine is useful to quantify the fraction of the cells which are live, which are apoptotic, which are late necrotic, which are late apoptosis process. But FACS machine cannot identify which are the genes are expressed more or which are the genes they are responsible for this apoptotic process. You understand what I am saying? So, therefore, QRT-PCR is an indispensable process in understanding the molecular level of understanding of the apoptosis process. Now, in the molecular biology literature, it is called gene of interest or GOI. GOI here is not Government of India, but GOI is here genes of interest. Okay. Now, genes of interest, as I said, that the first one is the BAX. BAX, BCL-XL, BCL-XS, these are like BCL2 family of genes, as I said earlier. Okay. Now, I told you, right, that what is called primer. These primers, they are forward transcription and reverse transcription. Why forward and reverse is required? Because RNA is your single strand, DNA is your double strand, right. So, one is forward means one is it will go this way, one is reverse means one it will go this way. So, what is the primer? As I said, primer sequence means GCC, that means one guanine, that will be followed by 2 cytosine. Then comes CTT, that means after 2 cytosine there will be 1 cytosine and 2 thymine. TTC, TSC, TTT, all those things. So, these are very specific to the Bax gene forward primer. Okay. Forward primer means here this one would be simply added. You understand what I am saying? So, one is forward, let us say this is forward and this is reverse. This is reverse means this is reverse here. Okay. So, this is the forward primer sequence, that means nucleotide sequence. This is the reverse primer sequence, that is the reverse nucleotide sequence. Okay. Now, this sequence is necessary to make the transcription, reverse transcription process from RNA to DNA in order to enable that process. Now, depending on the genes of interest, BCLXL, BCLXS, all those things you see, these are the primer sequence. FA1, you have the primer sequence, osteocalcin, COL1. Last two are beta actin, no, this is not B, beta actin and 18S, they are like housekeeping genes. Okay. In these housekeeping genes, you see that here also that primer sequence is different. If you notice, if you carefully notice that all these genes of interest, that Bax to up to 18S, their nucleotide sequence can never be identical. There must be some difference in certain nucleotide sequence. If you carefully go through it, you do not find any identical nucleotide sequence. So, this is that one this so, in the first cycle what will happen, if you have a double stranded DNA, then heat to separate the strands. So, if you heat it to little bit higher temperature like 50 degree or 70 degree, see these two double strand DNA, they will be separated. Okay. Then step 2 is what? Hybridization of the primers. Hybridization of the primers means those primers that we have discussed earlier slide, these primers can be added here. Now, stage 3 means that is the DNA synthesis from primers. So, here what will happen? This will replicate. So, S becomes S will produce another complementary strand S prime. If it is S prime, then it will make the another complementary strand S. So, here it is S and it is S prime in the step 1. Step 3, once these primers are added, then they will themselves will produce another complementary strand. In case of S prime it is S, in case of S it is S prime. So, the DNA synthesis is completed. Now, the other things that one can use that amplification of DNA using the PCR technique, polymerase chain reaction. Suppose you have a region of double stranded chromosomal DNA to be amplified. So, this is your S and this is your S prime. Okay. So, now I am showing you how to amplify only a small part of S and S prime to larger quantity, so that you can measure by polymerase chain reaction. Now, what you do, you can separate these DNA strands and add primer to that. So, separate means this is your small strand here and then you add green primer here and this is the red primer to other things. So, this primer would be of certainly would be of different nucleotide sequence. Okay. Then what will happen? It will produce another strand here, complementary strand. Now, this process from here, if you heat it to little bit higher temperature, as I have said here. So, again this DNA strands will be separated. So, black one will be separated from the green one. Again you can anneal the primer and then you can add this green and the red one. So, again it will form another strand here, complementary strand, and this process can be repeated, and so that from one double helix pattern of DNA you can end up having the so many number of DNA, replicated DNA with that particular genes of interest. So, if this single DNA helix here, whatever the expression of this final quantity, final number of this DNA would be much more, and that would be detectable, right. But in the process, what you see that there are couple of cycles here. This is the cycle number 1, this is the cycle number 2 producing 4 double stranded DNA molecules, and cycle number 3, this is the 8 double stranded molecules. So, 2 to 4, 4 to 8. So, and this is 1 to 2, 1 to 2 is the first cycle, 2 to 4 is the second cycle, 4 to 8 is a third cycle. Okay. So, similarly, you can have capital N number of cycles and large number of cycles for the expression of the DNA here. This is the other ones. So, now you can see that what is the typical amplification plot, that is the real time gene expression of these osteoblast cells. Now, these are like different genes of interest, that how they are amplified. This is your cycle number. You now know that what is the cycle that consists of. So, you can see the cycles can go up to 40 here. So, that is huge number of cycles. So, N is equal to 40. Now, these cycles, what you see, this is the reference values, and with increasing cycle number your genes of interest are actually amplified. All the genes of interest are amplified. Okay. Now, these genes of interest can be either osteocalcin or CBFA1 or different type of genes are there. In each group of genes, if you see that with increasing number of cycles, they are being more amplified. That means their concentration or their expression value is getting much and much more. Then coming to the BAX gene, BAX is that pro-apoptotic gene, right? Pro-apoptotic gene means these are like responsible for the apoptosis process. Now, this is with the temperature. Why temperature is required? You remember that you have the double strand DNA you want to amplify, you have to heat them, so that to separate the strand. Then you have to add some primer here. So, what is the temperature of this heat treatment with which, so you can see that in case of the BAX gene, it goes through a single peak, and this essentially tells you that this BAX gene is expressed in a natural manner, the way it should be expressed after the treatment with the hydroxyapatite 20% mullite eluates. Now, is this the quantitative values that I am giving, that CT is the cycle threshold value. Now, what is cycle threshold value? If you go back to this one, cycle threshold values for a particular genes of interest, for example, if it is the BAX or it can be the BCL-XL. Okay. Cycle threshold value means this is the, for example, your CT values. That means total number of cycles at which these particular genes of interest are expressed to the highest expression level. You understand what I am saying? What I am saying here, if you look at that, this is a BAX gene, this is, let us say, gene number 1, this is gene number 2, this is gene number 3. For the gene number 1, the cycle threshold value means, suppose this is the 14 cycles, beyond 14 cycles they are expressed to the highest possible level after the amplification process. So, their CT values is 14 here. If you go to gene number 2 here CT values are much higher, and it can be CT values is 32. That means, after 32 number of cycles, gene 2 will be expressed to its maximum amplified value. If you go through the gene 3, then here the cycle threshold value is 38, for example. So, that means here after 38 number of cycles, that gene 3 is expressed to its highest possible value. So, accordingly, for each of the genes you can find out that what is the typical CT values. Okay. So, for example, here it is the BAX gene, that is the pro-apoptotic gene. So, what you see, that cycle threshold value after 10% eluate concentration, it is 23.46, and delta CT value means this is your CT value is 23.46. Okay. I have mentioned here very critically, this is normalized with respect to beta actin housekeeping gene. So, what it means? This means, if you go back to this amplification plot, you have to follow it little bit carefully. So, this G1, G2, G3, these are the genes of interest, but at the same time you have that 18S gene and you have the beta actin gene, these are like housekeeping gene. So, these two genes, they can also be amplified with respect to certain cycles, PCR cycles. Okay. So, you have to continuously measure that what is the cycle threshold for the genes of interest and what is the cycle threshold for the 18S or beta actin housekeeping genes. So that you can find out that what is the normalized values means this delta CT values means that means this is the cycle threshold difference of the particular genes of interest, that BAX gene with respect to beta actin gene. So, it is minus 1.74 and typically the fold change is typically 2 to the power minus delta CT, and if you do little bit calculations it is coming out 3 to 3.35. Now, the question that should come to your mind, what is the significance of these values? Now, typically for your control or untreated cells, this 2 to the power minus delta CT is typically taken as 1. That means, delta CT values are 0. Why it is 0? Because whenever you are considering in that untreated cells, their beta actin expression will be the same as the normal cells. So, there will be no difference. So, there will be no difference between the beta actin and the BAX gene, it will be 0, and therefore, 2 to the power 0 is nothing but 1. Now, anything higher than 1 here, it is actually showing that more upregulation. So, this is not that much higher than 1. If you go back to this one, if you see that another BCL-XL gene and then it is compared or it is normalized with respect to 18S housekeeping gene. Now, here is that average CT value you see and delta CT values. So, average CT value means these are the number of cycles at which BCL-XL gene is amplified to the highest possible expression level. Okay. Now, when you are doing it with respect to the 18S housekeeping gene, it is coming out to be negative and the fold change therefore, it is going to be positive, that is that 4.71. But what you notice here that there is something going on at the 50% concentration level. Why something going on? It is going from 4 to 6 to 25. But again above 50% concentration, then your fold change values are going down. There is upregulation. Upregulation means that fold change values is typically greater than 1. 1 stands for control cells or control sample like untreated sample, and with respect to 1 it is severely upregulated, even it goes to 25. Now, if you go to that another one BAX gene with respect to the 18S housekeeping gene, similar analysis RT-PCR analysis, what it shows that it is very close to 1 and sometimes it is less than 1. So, that means BAX gene is down regulated. Down regulation and up regulation means how these genes are expressed in the treated cells with respect to the untreated cells. So, if it is down regulated, that means BAX genes are not that much active if it has to undergo very much highly apoptosis process, and typically these analyses are done after 6 hours treatment. Okay. So, that means the cells are not really down regulated, cells are not really undergoing severe apoptosis process. Now, BCL-XL to BAX ratio actually determines what? It determines the ratio of the anti-apoptotic gene to the pro-apoptotic gene. So, this is that anti by pro-apoptotic gene, because BCL-XL values after 10% concentration is 4.01, 25% concentration is 5.03, 75% concentration is 7.41 and 100% concentration is 7.94. So, what you notice, if you go from 10% to go to 100%, your BCL-XL to BAX ratio is just doubled, almost doubled, 4.01 to 7.94. So, what is the meaning of that? It is meaning of that is that this hydroxyapatite 20% mullite concentration, if it is increased from 10% to 100%, the BCL-XL anti-apoptotic to pro-apoptotic gene ratio, it also increases twice. That means there is increased BCL-XL to BAX ratio. What it means? It means there is more expression of anti-apoptotic gene with respect to the pro-apoptotic gene. That means the cells will not undergo normal apoptotic process if you increase the concentration from 10 to 100% simply because of the abundance of the anti-apoptotic gene, which will restrict the cells to undergo the normal apoptosis process. Is it clear? So, for any kind of this RT-PCR analysis with the cells, one has to always determine the final values of the BCL-XL by Bax ratio, because that is the anti-apoptotic to the pro-apoptotic gene expression ratio. Now, there is some other genes which is also known as the CBFA genes. Now, CBFA genes are essentially responsible for the tissue formation. Now, if it is CBFA genes are more than one, as the way it is here, you can say that the CBFA genes are upregulated, and therefore, these hydroxyapatite 20% mullite eluates, they will essentially induce the more tissue formation. There is another genes also known as that osteocalcin genes. Now, osteocalcin genes are mostly responsible for the bone mineralization process, but osteocalcin, what you notice here, this is down regulated, or this is insignificant effect, because for the control cells like untreated cells, as I said, your fold change is 1. So, with respect to 1, it is hardly 1.05, 1.13, it is, there is not much difference. So, the control cells osteocalcin expression is same as the osteocalcin expression in this different eluate concentration. Okay. Now, this is the another gene is known as the collagen gene expression. Now, in the collagen genes, what you notice here, the fold change is little bit higher than 1, but it is certainly this fold change is not greater than 5 or something like that. So, there is insignificant effect on the fold change values in the case of the collagen genes, and therefore, this is the ECM protein. Collagen is nothing but the extracellular matrix protein. So, these particular eluates will not have much effect in terms of the collagen synthesis also. So, now you can see that what are the different mechanisms. So, ultimately this is the last slide, that possible mechanism is that hydroxyapatite mullite, hydroxyapatite is Ca10 PO4 whole 6 OH whole 2, and mullite is alumina and silica. So, 3 alumina and 2 silica. So, what is happened in case of this eluate treatment, your calcium plus 2 ions are there, your phosphate ions are there, and silicon ions are there, and Al plus 3 ions are there. So, these calcium ions can go inside the cytoplasm, some calcium ions can go outside of cytoplasm also. But what is we propose that silicon ions they are getting in and they are causing the damage to the double helix pattern of the nucleus in the DNA, and that is causing the more toxicity or more cell death when you are treating the hydroxyapatite mullite particles. Okay.